The last 25 years of the Institute has defined the character of the Institute. And it's based on our ability to go after diverse ideas in diverse systems simultaneously. The work that has come out of our laboratories on the fields of memory, developmental biology, stem cell biology, and genetics has opened the door to avenues that were deemed impenetrable before, thanks to the generosity of our founders, Jim and Virginia Stowers.
Stem cell niche is really like the real estate, location, and location. You move away from the particular location, your value is reduced. Same thing true for stem cell. If stem cell leave the niche, it's no longer a stem cell. We're looking for adhesion molecule, like enter. The stem cell cannot easily move away. Stem cell can continue cell renewal.
My lab worked 25 years in the Stowers. We first discovered the stem cell niche in the bone marrow that helped understand the concept of a stem cell niche. What is the niche, including which cell type? What molecule engage the stem cell with niche interaction? Stem cell also have the interaction with their microenvironment niche, particularly immune cell. They can somehow manipulate the immune cell. Instead of attack cancer stem cell, the immune cell start to protect the cancer stem cell.
The gene family that I studied, called HOX, are very important in brain development. Could we find new cues or new signals that were sending instructions to them? And we found several. When we modulated this, it not only affected brain development, but it made bones stronger in a mouse model. Now, if I was in another university, I probably would have had to talk to venture capitalists and started my own company to do this. Jim was able to say, why don't you let us develop this? And they licensed it to Amgen. 15 years later, it ended up being a blockbuster drug for osteoporosis.
When I moved here, I already had discovered the segmentation clock. At the time, there were oscillators belonged to the world of physics, and so we were bringing this to the biology. It embodies some sort of elegance, to pattern an embryo with an oscillator, a system that reproduces itself like a clock. It caught many people by surprise. There was a model proposed 25 years before. This model was completely forgotten as something non-biologically plausible. And so here we were really showing that not only it was plausible, but it existed.
The research is about how aneuploidy, which is the state of having unbalanced chromosome numbers, can profoundly impact the behavior and property of the cell. So under stressful conditions, certain aneuploidy cells can be selected to become stress resistant. This genetic diversity brought about by aneuploidy can allow cancer to become resistant to a variety of insults such as chemotherapies or immune cell surveillance. There are a lot of thinkings now in cancer research field based on our very basic fundamental discovery.
So I started out trying to understand how you create memories and keep it, some cases, your whole life. The solution that emerged is actually something that we knew about it for a long time, but in a very different context. The proteins can aggregate, and when they aggregate, they can create a stable substrate for memory. What makes them sometimes to make memories and sometimes interfere with memories? If we really understand these questions, I think that's going to provide the solution how to treat this disease, such as Alzheimer, Parkinson, Huntington.
Well, one of the things that we discovered at the Stowers Institute is that when RNA polymerase is making an RNA copy of a gene, it has to move down the length of the gene, and it causes damage to the packaging proteins, mainly histones. And we found an enzyme, a histone methyltransferase, that binds to RNA polymerase, moves with it, and then puts marks on the histone proteins along the gene, which are then used by other proteins to come and repair the packaging behind the RNA polymerase. So this is a very fundamental thing that happens on every gene, in every cell, in every eukaryotic organisms, from yeast to humans, which hadn't been known before.
We identified that when you make these translocations, although the translocations are on different chromosomes, the product all end up in the same complex in the protein. and that's super elongation complex. That basically solidified that transcription elongation control is a key regulatory step for development. And if you make mutations in elongation factors, you can get cancer. Polymerase is moving along DNA template. The rate at which it's moving is highly regulated.
We focus on trying to understand the mechanisms that underlie meiosis, the process that creates the right number of chromosomes in both sperm and eggs. Imagine that the structure we're working with, it has a long Greek name, the synaptodemal complex.
This work is about the function of Hox genes in the sea anemone Nematostella vectensis. We showed that the genes and molecular pathways that help build the adult organism in sea anemones, corals, and other cnidarians is fundamentally similar to what builds our own body plans in humans and other vertebrates. We're sort of probing the evolutionary history of animals. So absent a time machine where we can go back and see what animals looked like 700 million years ago, we're using our studies of cnidarians to really triangulate back in time and understand the ancient biology of the common ancestor of all complex animals. I also hope that long-term, our efforts at technology development and tool development for cnidarians can transform the study of corals, which are under threat.
One of our laboratory's major findings was the identification of a stem cell in an organism known as planarians that appeared to be responsible for producing all of the cell types present in this organism. Technologies that were available to us here allowed us to purify these stem cells and watch them in action as they rescue the regenerative abilities of animals, planarians that had lost that ability. We had now begun to bring into the lab organisms that ordinarily had not been studied for these processes, among them the snail, hemichordates, and an African killifish. All of these animals can regenerate if we can begin to identify common denominators that will tell us that regeneration was something that was ancestral and somehow was kept in some animals but lost in others.
In collaboration with Anshul Kondage's lab at Stanford, we developed BPNet, which stands for Base Pair Network, a neural network that makes a connection between DNA sequence and genome-wide binding patterns that we had experimentally measured in mouse embryonic stem cells. The model learned general rules for the cell type that were predictive, and it also uncovered new patterns that we hadn't expected and that we were able to experimentally validate. And this is fantastic because it does show that the so-called code, the language of the DNA, is solvable in principle, and also that we can discover new mechanisms of gene regulation using this approach. The Holy Grail is to be able to read all the health information that's written in our genome. This would uncover information on how to keep healthy, on how to treat diseases in a more personalized way, and so I think it would be huge.
We've been working on a type of chromosome called a Robertsonian chromosome. It's a type of chromosome that forms when two chromosomes fuse together. They've been recognized for over 100 years, but we've never understood why they form. Due to recent advancements in technology, we've been able to find out the specific regions of those chromosomes that fuse and how that fusion happens. They can cause various health issues like infertility and developmental syndromes like Down syndrome. So it's important to understand how they form. And since this is a mechanism of chromosome changes and speciation, it really gives us an insight into how chromosomes are part of that evolutionary process.